I'm just going to really quickly walk through this, and I'm going to show you this cool animation of PCR that I think you guys will really get a kick out of. So the basic idea is if you have one single copy of double-stranded DNA, and you want more than one copy, whether it's for molecular biology purposes, whether it's for forensic purposes, all you need to have is a little bit of information about the area of the DNA you want to copy. So what PCR generally is used for is not amplifying all the genome of the individual, though there are techniques for whole genome amplification that I've never done myself, but they've sort of continued to innovate around PCR. Generally what you're doing is you're amplifying just a little teeny section of DNA. So all you need to have in terms of information for doing PCR is the sequence that flanks the area that you want to amplify. So that's the information you need in order to do PCR. So you need information about the sequence here and the sequence here. And what you do is you design these short little sections of DNA that are called primers. And they're sequences of about 15 letters or so that are complementary to the region on either side of the area that you want to amplify. So do you guys remember from the talk we went to on telomeres and telomerase, how DNA doesn't start copying from an area that is single-stranded? It has to begin copying at an area that's double-stranded. Well, when you're using this technique, DNA polymerase, you're using the enzyme when you're using this technique, polymerase chain reaction, you're using the enzyme DNA polymerase. And it's unable to start and initiate copying at an area unless it's double-stranded. So what you do is you take your DNA, you mix it together with these short little sections that are complementary on either side of the area you want to amplify, and you let them hydrogen bond to that spot. Included in the mix is this orange sphere here, which is the DNA polymerase, which is going to latch on to that double strand, and when you heat it up to the appropriate temperature, it ratchets its way along and makes a complementary copy. So what you do is you mix all this stuff together, you heat it up to denature the double helix, you then cool it down slightly to attach the primers, and then heat it up a little bit more to let the enzyme become very active, and it skitters its way along the DNA, making a copy. So you've now gone from 1 to 2 copies. So then you simply repeat. You heat it up so it denatures, you cool it down so the primers stick, you heat it up a little bit so the enzyme becomes active, it cruises along and makes a copy, generating 4 total copies. So you get this geometric progression of 1 to 2 to 4 to 8 to 16 to 32 and so on, taking you from a teeny speck of blood to buckets full of DNA. Yeah?
So would this be happening in like a solution that has all the precursors to create DNA present in it? Because I mean, I don't feel like polymerase would be able to just manifest.
Absolutely. So you're totally right. And the one thing I didn't mention is you add into the mix a whole bunch of nucleotides, A's, T's, C's, and G's.
So it can have like things to grab from and like place on the ladder. Mm-hmm. Okay.
Exactly. So let me show you this cool little. Prepare for your minds to be blown. Yeah, we have sound effects, guys. It's true. So I'll let you read these as I walk through it. Oh. No wonder you guys are looking at me so strangely. There we go. Can you see it now? just getting you guys excited for it. So PCR is able to take a single copy and make billions and billions of copies from it. And so what I'm going to do is I'm going to take you through that process. So imagine that you leave a hair at the scene of a crime, and we want to extract DNA from that and figure out who you are. So I'll take my DNA. So this is a setup just like you guys have been working with today. You've got your ice buckets, you've got racks with these Eppendorf tubes in it, and I'm going to take a pipette, look at that, and I'm going to pipette DNA and move it over into my reaction tube. So I'm going to first take my DNA, it's a really fun game. Did you notice that I ejected the tip and put a new tip on? So that's my DNA is now in the tube. Now I'm going to take the primer. That's the complementary short sequence on one side of the area I want to amplify. Put it in the tube. New tip. Primer 2, it's on the opposite side and flanks the other region we want to amplify. Now I have to add those nucleotides that Tyler asked about. So these are all the A's, T's, C's, and G's to make DNA. So the last thing we're going to do is we're going to add DNA polymerase. And now when we heat this up hot enough to denature the DNA, we actually heat it up to about 95 degrees Celsius, which is basically boiling water temperatures. And so typically we would think enzymes would be what at boiling water temperatures? Killed. What's the term for that? Denatured, right? We'd expect the enzyme, the protein, to be denatured at high temperatures. So what we use is a specialized polymerase that's extracted from or isolated from a species of bacteria that lives in hot water vents. And so it's actually evolved changes in the polymerase that make it resistant to being denatured at high temperatures. So I take my DNA polymerase, put it in the tube. Now I'm going to put it into this magic machine called a thermal cycler, or you can just call it a PCR machine. I play this game all the time. It's so fun. It's amazing because I win every time too. So I'm going to click start, and we're going to walk through what happens inside of the tube. So this is the target sequence, the area we want to amplify. So the first thing I do is I denature it by heating it up to boiling water temperature. Now I cool it down to about 50 degrees or around there, and that allows a process called annealing, when the primers stick on to the double strand or onto the single strands of DNA to occur. So do you see the primers attaching here and here? I'll move on, and I'll heat it up hot enough that the polymerase becomes active and make the copies. One cycle is complete. I just denatured it. Do you see how the double strands separated from each other? I'm going to anneal. And then the next process is called extension or extending, when the polymerase moves along and extends beyond the primer and adds the complementary sequence. So now I have 4 copies of the information. You guys see that? Move on to the next cycle. So they make a point of showing you that now at this point we've actually made areas that are just the fragment you're interested in amplifying. But from our perspective, we're just interested in getting copies of the DNA in total. So if I do it one more time, I double the total, double it again, double it again.